Ashwini. Um, we'll wait for a few more minutes to see if some more people are joining and then start today's session. Good evening, Ashwini. I hope I was audible the previous time. Uh, we'll just wait for a few more minutes and see if some more people are joining and then start today's session. And also, please let me know if the screen is visible and if my audio is clear. Am I audible? Good evening. We'll start in a couple of minutes, like around 7:15 we'll start the session. Please let me know in the chat box if I'm audible. Okay, so good evening and welcome to the live practice session, week 5 for the NPTEL course on environmental biotechnology. I am Guru Priya, Prime Minister's Research Fellow in the Department of Civil Engineering, IIT Madras. So, in today's session, we will see few practice questions uh, from previous week as well as this week's NPTEL lecture. So last week we were seeing many questions, and we there were also few more questions that we could have discussed, and because of time constraint we stopped. So just from the next question that I was having, we will start the discussion today. So as we were doing in the previous 4 sessions, I will have some question and answers, which I'll give some time for each questions for you to answer in the chat box, and after that we can discuss it. The first question is an assertion and reason question. So the assertion is, fungi are critical in composting due to their ability to degrade polymers, and the reason is fungal hyphae secretes extracellular enzymes that may break down xenobiotic compounds. So you can just choose the right answer. Okay, so first of all, composting is one of the solid waste management techniques. So it is very much related to the environmental biotechnology that we are talking, because that is one of the biological waste treatment processes. So in composting, what ideally happens is we have this organic fractions of the solid waste that we generate, which are biodegradable, and we make them as a heap, and we have some inocula or some bacteria to degrade them. Uh, so the inoculum is not only bacteria, it can consist of other microorganisms also. So in that case, fungi is also a organism that can be used in composting. So what happens is, uh, the fungi has some hyphae. So hyphae is a thread-like filamentous and tubular structure, uh, and it is the vegetative unit of fungus. So like you can imagine something like a root. It is not exactly a root, but it is filamentous and tubular. um, so these fungi are generally very much capable of degrading polymers, and in any solid waste treatment process, uh, we need extracellular enzymes only. Only these enzymes can be very efficient, because all these polymers are large biomolecules which cannot enter the cells of the microorganisms. So first of all, this breakdown of the polymer into simple molecules happens outside of the organism. So that is why extracellular enzymes are very much needed, and this fungal hyphae is a very good medium for the synthesis and secretion of extracellular enzymes. So the organism synthesizes the enzyme and then sends it outside the cell. It secretes it outside the cell. And then these compounds can break down the xenobiotic compounds. So when these large polymers, when they are broken down, all these simple nutrients are taken inside the cell and further they are processed, like other fermentation or things happen, leading to um the conversion of the waste into some other resource, and finally the compost is ready for use as manure. So this is the whole process of composting. And xenobiotic compounds generally mean a foreign substance. So any foreign substance is a xenobiotic compound. So this extracellular enzymes can break the xenobiotic compounds that is present in a organic solid waste. So based on the explanation, the answer that you have given is exactly right. Um, Fungi is critical in composting because of its ability to degrade polymers. That is also true. Fungal hyphae secretes extracellular enzymes that break down xenobiotic compounds. That is also true. And just because fungus can efficiently secrete extracellular enzymes, they are critical in composting. So the reason is correctly explaining the assertion. So in that case, A is the right option here. So the next question is also an assertion reasoning question, where the assertion is algae contribute to BOD reduction in wastewater by assimilating organic matter. The reason is all algae produce oxygen via photosynthesis that supports aerobic bacterial metabolism. So you can think about both the assertion and reason, and then you can give your answers. Okay, so we have A as answer and B as answer. Let's see which is right. So primarily most algae are photosynthetic, right? So what happens is they take up inorganic carbon, that is carbon dioxide, and then fix it and release oxygen. So that we all know. So the reason states algae produce oxygen via photosynthesis that supports aerobic bacterial metabolism. So that statement is true. So we all know that algae is photosynthetic, so it can take up carbon dioxide and release oxygen, and it supports aerobic bacterial metabolism because it releases oxygen and increases the dissolved oxygen level. So when we take up any wastewater system, so this question is primarily focused on wastewater system. So in a water system, algae produces oxygen, therefore it increases the dissolved oxygen content in the water, and hence the aerobic microorganisms or aerobic bacteria present in the water can utilize the oxygen and further uh, do the conversion of the organic matter present in uh, the wastewater. Whereas coming to the assertion, algae contribute to BOD reduction in wastewater by assimilating organic matter. So there is we all know that it is photosynthetic and the carbon source is going to be carbon dioxide and not the organic matter present in the wastewater. uh, Algae like indirectly contributes to BOD reduction. So BOD is biochemical oxygen demand, and it is a direct indicator of the organic pollution in the wastewater, right? uh, so can anyone explain what exactly is BOD? I'll just wait for 1 minute. I'm sorry, I lost the connection. Um, so BOD is the biochemical oxygen demand. So what it essentially So what BOD essentially means is that it is an indicator of the organic pollution in wastewater. How when we do a BOD test, we tend to find out what is the oxygen requirement for decomposing the organic pollutants present in the sample. So that is biochemical oxygen demand. So what we essentially do is the measurement of dissolved oxygen in the water. Initially what was the oxygen present, and after 5 days, or BOD 5 is generally what we measure. So after 5 days, what is the dissolved oxygen content? Based on that, what is the oxygen demand for the conversion of the organic pollutants is what we call as BOD, right? So BOD, high BOD indicates large amount of organic pollution. When there is high demand for oxygen, there is more pollution, and when there is less demand, there is less pollution, or less level of organic pollution per se in that matter. So algae contributes to BOD reduction. Yes, true. Algae is contributing to BOD reduction in wastewater. But how it is contributing? It is contributing by increasing the dissolved oxygen in the water and supporting the growth of aerobic bacteria, but it does not directly assimilate organic matter. So the assertion is wrong here. I hope I am clear. I am repeating. BOD is an indication of organic pollution, and algae is contributing in the reduction of BOD, which means it helps in reducing organic pollution in wastewater. But the mechanism by which algae contributes to BOD reduction is very indirect. How? Because algae are photosynthetic, they produce oxygen and support the growth of aerobic bacteria. These aerobic bacteria consume the oxygen and aid the conversion of this organic pollutants into like simpler forms. Okay, but algae does not directly assimilate organic matter. That is very rare, and maybe some mechanisms like osmotrophy or something are involved in that. In majority of the cases, algae only contributes by indirect methods and not by direct assimilation of organic matter. So the correct answer is A. Assertion is incorrect, but the reason is correct. I hope this answer is clear. If you have any doubts, please post in the chat box. I will again give the correct explanation. Okay. In the meantime, I will post the next question. Now, first we saw about fungi, then algae. Now we are specifically seeing about yeast. So the assertion is yeast growth always produces hyphal mycelium. The reason is yeast are single-celled fungi reproducing by budding or fission without hyphal. I think this question is a little bit easier. I'll just wait for a couple of minutes for you to answer this. Exactly. So yeast does not produce any hyphal mycelium. So yeast is single celled. It is also a fungi. It is single celled and it reproduces by budding or fission. Budding means an extra growth in the cell which finally withers off from the original parent cell. And fission, as we all know, is a direct cell division. So yeast can reproduce, yeast is single celled and reproduce by budding or fission, and it does not form any hyphal mycelium. Again, a very simple question, like to reiterate the previous question, you can answer. Filamentous fungal cells form networks called. Okay, the answer for this question is mycelium. But this conidia part that you had given in response to the previous question, conidia is generally for molds and it is for filamentous fungi, and those are spores of those fungi, and we do not talk about conidia in case of yeast generally. Okay, so we'll move on to the next question now. So which pigment distinguishes chrysophyta from chlorophyta? So for this question, we are talking about two algal types. One is the golden brown algae and the other is the green algae. So we know that these two algae are going to be of different colors and both are eukaryotic algae. So apart from their pigment differences, they have differences in their cell wall, food storage and as well as the habitat that they are present in. So we have two types of eukaryotic algae, Chrysophyta and Chlorophyta. As your answer suggests, the main difference can be between the pigments, either chlorophyll or fucoxanthin. We are not worried about phycoerythrin or beta carotene here. So in case of Chrysophyta, it is the golden brown algae. Uh, Whereas the Chlorophyta is the green algae, but both these contain chlorophyll. In Chrysophyta, we have chlorophyll A and C, and in Chlorophyta, we have chlorophyll A and B. So chlorophyll B can be the answer, but the distinguishing factor is the presence of fucoxanthin. So if it is going to be chlorophyll A, B or C, all of it is mostly going to give a greenish color to the algae. But the pigment that distinguishes Chrysophyta and Chlorophyta is fucoxanthin, because fucoxanthin is the one that makes us term Chrysophyta as a golden brown algae, and the lack of fucoxanthin makes chlorophyll the dominant pigment in Chlorophyta and makes us term it as a green algae. Though chlorophyll B is also different in both Chrysophyta and Chlorophyta, like only one has chlorophyll B whereas the other doesn't have. Fucoxanthin is the pigment that makes us distinguish both these algae significantly. So in that case, the most suitable answer for this question is going to be fucoxanthin. The next question is about Euglenophyta. How do we differentiate Euglenophyta from Cyanobacteria? Exactly. So Euglenophyta is eukaryotic, whereas Cyanobacteria is prokaryotic. Euglenophyta has nuclei and other membrane-bound organelles which are not present in Cyanobacteria. So exactly, all your answers are right. So the next question is: a high abundance of amoeba in wastewater indicates what? right answer for this question is high BOD, because low organic content or high dissolved oxygen is not the answer, but right, high BOD and low microbial diversity can be the answer. The low microbial diversity might be because of high BOD, like because of unfavorable condition that gives pressure for the microorganisms, and hence the diversity may be reduced. But the direct related answer for this question is a high BOD. So generally, when we talk about high amoeba, it means that there is a shock load of BOD, which means suddenly there is a high influx of BOD in the wastewater. So that is why we have large amount of amoeba, and high BOD suggests there is low disorder. So that way C is not the right answer, and high BOD means the organic content in the wastewater is high. Microbial diversity is very indirectly related to high BOD, but we are talking about what the presence of abundant amoeba indicates, that is influx of shock loads of BOD or high BOD, because amoeba can also have some outer protective shell that helps its survival in uh, very high BOD wastewaters. So B is the right answer. The next question is, helminth removal in wastewater is challenging due to which of the following options? So helminths are parasitic worms, so they are of very small size, and generally their filtration is difficult. Uh, so cyst hydrophobicity, two of them have given the answer. What is cyst hydrophobicity? Is a tendency of the cyst to repel water. So this makes them uh, very difficult to adhere to surfaces during wastewater treatment. So because of this, and also they have a small size contributing to hard removal. So when talking about the other factor about the cyst hydrophobicity, cyst formation essentially is during some adverse conditions that helps the helminth to survive for a longer time. So it can stay dormant and then it can also come back to its very active life form. So their small size is true, but if only the small size were a problem, we could have gone for some advanced technique, because we are even removing um, materials from wastewater which are still more smaller in size than the helminths. So their small size is not that much challenging, but the cyst hydrophobicity contributes in two ways. One is the cyst is of very small size and the removal is difficult. The other thing is this cyst can stay dormant, and then it can also come back to its active form. So in that way, cyst hydrophobicity is the major challenge in helminth removal from wastewater. The next question is identify the true statements out of the following. The first statement is average carbon content in microbial biomass is 70%. Culture medium is used to grow, transport, and store microbes. Chromium, cobalt, calcium, and copper are micronutrients for microorganisms. And nutrient broth is used for the cultivation of fastidious bacteria. So, which of these statements are true? Okay. Uh, so the average carbon content in microbial biomass is 70%. It is not the average carbon content, but mostly the water present in microbial biomass is 70%. So that statement is false. Culture medium. Uh, so culturing is growing some microorganisms in a laboratory setup, and for that we use some medium for growth. So that medium essentially provides the nutrients that are required for the growth and survival of that microorganism. So that is the culture medium. Culture medium is used to grow microorganisms, transport them, and store microorganisms. That statement is true. So the next statement is chromium, cobalt, calcium, and copper are micronutrients. So in this question, chromium, cobalt and copper are micronutrient, but calcium is not a micronutrient. It is a macronutrient. So it is required in larger amounts. So it is also called as macro mineral, and it is required in larger amounts for prokaryotic organisms like bacteria, because cell wall structure and some signaling, everything is dependent on calcium in microorganisms, and hence calcium is not a micronutrient, whereas chromium, cobalt and copper can be accounted as micronutrients. So that statement is false. Nutrient broth is used for cultivation of fastidious bacteria. So fastidious microbes are those that are difficult to grow in labs, because they require complex nutrition and environmental conditions. So simulating that complex nutrition and environmental conditions in laboratory setup is very difficult. Uh, but we kind of do it using like specialized culture medium, not nutrient broth. Nutrient broth is a very basic medium, and hence it cannot be used for the cultivation of fastidious bacteria. So that is also false. So the answer is only B here. C is false because calcium is also listed in the option C. Calcium is a macronutrient. So the next question is again a assertion and reason question. Define, as the assertion is, defined media are composed solely of compounds with known chemical formulas. The reason is complex media are preferred for autotrophs because of their precise nutrient uh, needs are well understood. Defined media is composed of compounds with known chemical formula. That is why we call it defined. Complex media, we don't know the exact chemical formula or chemical composition of the complex media, and that is not preferred for autotrophs, because that is preferred for autotrophs, but complex media essentially means we do not know the exact composition of the media. Therefore, the assertion is correct here and the reason is incorrect. The next question is about auxotrophs. Auxotrophs cannot grow on minimal medium lacking specific growth factors because auxotrophic strains lose biosynthetic pathways when those metabolites are abundant in nature. Okay, so the assertion is essentially the definition of auxotrophs. So, what are auxotrophs? Auxotrophs are organisms that lack some biosynthetic pathways. So, they cannot produce either certain amino acids or vitamins because they lack that biosynthetic pathway. These are essentially mutant organisms. So they have been mutated in such a way that they cannot produce a certain amino acid or vitamin or some nucleotide. So we have to supplement uh, that particular factor with for the growth of that auxotroph. But what we do in a minimal medium, we only supplement uh, essential carbon source, water and inorganic salts, but not the other growth factors. So in if the minimal, in addition to that minimal medium, if you are going to add that particular growth factor which the auxotroph cannot produce, then only the auxotroph can grow. So the assertion is right. And the reason states that auxotrophic strains have been mutated, which means they have lost the biosynthetic pathways when these metabolites are abundant in nature. So when we are going to supplement excess of a particular amino acid or a nucleotide or some vitamin, they essentially lose the biosynthetic pathway through which it has to be produced and become auxotrophic. So both assertion and reason are right, and the reason is the correct explanation of A. So the assertion is RNA content per cell increases with high growth rate and RNA content of bacteria is lower than fungi. sorry for I lost my connection in between. So the next question, the assertion is RNA content per cell increases with high growth rate and RNA content of bacteria is lower than fungi. If anyone had noted the question and answered before, please post the answers again, because as I lost my connections, all the previous chat also got deleted. I'm sorry that even the screen sharing was stopped. I hope it is visible now. So growth rate is the rate at which the cell multiplies or grows. So generally we don't talk about cell size increase or something when we talk about growth rate. We essentially talk about the multiplication time. So in how much time one cell can produce its offsprings, that is growth rate. So when the growth rate is higher, the RNA content per cell also increases, because the entire metabolic process is going to be very fast. Therefore, the RNA content per cell increases with higher growth rate. And this RNA content does not talk only about mRNA, tRNA, or rRNA. It entirely consists of all the different types of RNA that your cell can have. So in that case, the assertion is correct. When we have higher growth rate, the RNA content per cell is also higher, or it increases. The RNA content of bacteria is lower than fungi is not correct, because the growth rate of bacteria is generally higher than that of fungi. Bacteria multiplies in a very shorter time than fungi. So therefore it has a higher growth rate. As we all know that the assertion is right, when bacteria has a higher growth rate, it has higher RNA content than fungi. So that is why C is the right answer. Uh, the assertion is right, whereas the reason is not. So the next question is, storage compounds supply energy and precursors during nutrient scarcity, where the reason is storage lipid is a storage compound present in both prokaryotes and eukaryotes. Storage compounds essentially means when there is some excess nutrient present, like it may be any mineral like phosphate or some other compounds like lipid. Storage compounds supply energy, and they can act as precursors during nutrient scarcity. So we have it stored, and when we need, we are going to use that compound. So it can include things like starch, glycogen, lipids, or phosphates. Many things can act as storage compounds. Storage lipid is a storage compound. Yes, present in both prokaryotes and eukaryotes. So this is the catch here. Storage lipids is not present in both prokaryotes and eukaryotes. It is present only in eukaryotes. So that is why assertion is right and the reason is incorrect. It is not present in both type of organisms, but present only in eukaryotes. So that is why the reason stated here is incorrect. If uh, if it had been given rightly, then only the answer is B. Both A and R are correct, while they are not explanatory. So the next question is, which pathway diversion yields biosynthetic precursors at the expense of ATP? So here we are talking about which pathway diversion. So pathway diversion means some alternative pathway that we are using. Complete oxidation, anaerobic fermentation are like not alternative pathways or diversion pathways. In case of electron shuttle to terminal electron acceptor, um, it is not at the expense of ATP. We are talking about a diversion or alternative pathway which happens by utilizing ATP. So that is why partial oxidation um, with intermediate withdrawal is the pathway diversion that can yield biosynthetic precursors by utilizing ATP. So this is not a very common phenomenon, like when the nutrient availability is very high, this pathway diversion does not happen. Only at very in need, only at times of high need, this partial oxidation with intermediate withdrawal happens. So this intermediate is the biosynthetic precursor and it is produced by utilizing ATP. So next question, culture medium solidified with agar is used for. I think this is a very easy question. plate cultivation?
రాత్రి 11 అయింది వచ్చినాం. ఫ్యామిలీతో రావచ్చా, అక్కడ జనాలు ఉన్నారు ఫుల్ కన్నా. ఎక్స్ప్రెస్ రాలని జనాలు ఉన్నారు. వచ్చేస్తారు, ఇప్పుడు వచ్చి రాలేదు కానీ వాళ్ళకి వచ్చేస్తారు.
The next question is uh, which factor does not significantly vary with growth rate? Exactly. DNA content does not significantly vary with growth rate. It remains stable with growth rate. Like the protein content, RNA content, and ribosome content varies with growth rate, whereas DNA content remains stable even when the growth rate is higher or lower. So, from the following statement, identify the false statement. resources for metabolism and selective pressures for replication and heredity. So that is true. Environmental factors allow the use of certain metabolic pathway. So it decides which kind of metabolism is used. Therefore, it produces the resources for the kind of metabolism to take place. And it also provides a selective pressure, like it directs the uh, microbial evolution. The environmental factors also direct the microbial evolution and provide selective pressure for the replication and heredity of microorganisms. Some kind of metabolism is constantly turned on and it get passes on to next generation, whereas some genes get turned off. So all this happens in case of replication and heredity. So I'm really sorry that the network is really unstable from my end today. I'll try to rectify it in the next week. Uh, Please adjust for this week. So the environmental factors essentially provide the resources and selective pressures and direct the microbial community dynamics. In case of chemosynthetic organoheterotrophs, they allocate more genes for amino acid and carbohydrate metabolism. That is true because they rely on chemical synthesis and organic matter. So we know that heterotrophs rely on external carbon and energy source. So they are chemosynthetic. So they allocate more gene for amino acid and carbohydrate metabolism, which in turn is the major biomolecule required for cellular structure and function. That is also a true statement. Whereas in case of C and D, prolonged exponential microbial growth occurs commonly in natural habitats. No, uh, we cannot see prolonged exponential microbial growth. Exponential growth in the sense uh, we see an exponential increase in the growth rate. So when we talk about this microbial metabolism, we have a lag phase, we have log phase and stationary phase, and then decline phase. So the lag phase is a straight line, which means the organism is getting adapted to the new environment, and then comes the logarithmic phase or the exponential growth phase. So the growth in this phase is exponential or logarithmic. uh, in natural habitats, the resources are not they are not very uniform, so you cannot see a prolonged exponential microbial growth phase in that system. Based on the resources or nutrient availability, the exponential phase also can be seen. So prolonged exponential growth phase is not seen. Dormancy in microbial populations is a response to continuous nutrient abundance. So here, up to the last word, everything is fine in this statement. It is not a result of nutritional abundance, but nutritional scarcity. Whenever there is a nutrient scarcity, then the microbial population goes into a dormant state. Dormant state in the sense it does not have active metabolism. So whenever again the nutrients availability is becoming abundant, it comes to a active state. So dormancy in microbial population is a response to continuous nutrient scarcity and not abundance. So that is why D statement is also false here. So the next question is, microorganisms may switch from autotropy to heterotropy based on environmental conditions. The reason is genetic versatility enables adaptation to fluctuating resource types. So the answer is A here. Microorganisms can switch from autotropy to heterotropy based on the resource availability. This is because they are genetically versatile and can adapt to fluctuating resource types. So the next question is, photosynthetic autotrophs de-emphasize inorganic ion transport genes because they synthesize organic compounds independently of external inorganic ions. photosynthetic autotrophs. So autotrophs which rely on photosynthesis de-emphasize inorganic ion transport gene because they synthesize organic compounds independently of external inorganic ions. So we all know that photosynthetic autotrophs, they are going to synthesize organic compounds, and this synthesis is independent of inorganic ions. Uh, Inorganic ion channels are mostly not used in the entire photosynthesis process, so that is why they de-emphasize on this inorganic ion transport genes. So this is a direct question where both the assertion and reason are right, and the reason is the correct explanation of A. So the three critical traits of life discussed in physiological ecology are, we saw in one of the previous questions. Okay. Uh, so based on the lecture, it was metabolism, replication, and heredity. Reproduction, metabolism, yeah, replication or reproduction, metabolism. Uh, Instead of homeostasis, it is heredity. So the interaction between dash and dash over evolutionary time is referred to as dialogue of selective pressure. So the answer is the interaction between the habitat and microorganism over evolutionary time is referred to as dialogue of selective pressure. So the habitat in indeed species and their environment. So the habitat essentially has the resources and other conditions required for growth, and there is some microbes present. So depending on the abundance or scarcity of uh, this resources or the conditions, the evolution happens. Okay, so the next question is, dash in habitats result in molecular, metabolic, and physiological diversity. So all the fill in the blank questions are like open-ended questions only, just to make you think from no options or anything, just from a very contextual point of view. So if you're not giving the exact answers that I have as answers, but nearly the same, then it's also completely fine. So I'm framing the question and answer from my perspective. From your perspective, if the answer is more or less close, then it's completely fine. Okay, selective pressure generally results in molecular, metabolic, and physiological diversity. So the next question is about R selected and K selected growth strategies in microbes. What are R selected and K selected growth strategies in microorganisms? Just give few keywords like one line answer. So yes, are selected species, they can grow and reproduce rapidly by exploiting the resources in uncrowded habitats, while K-selected species conserve resources and grow slowly in dense environment. This is the difference between R-selected and K-selected growth strategies. R-selected species can grow in uncrowded habitats, whereas K-selected species grow slowly in dense environments. Okay, so the next question is, which organism type oxidizes inorganic compounds to derive energy and fixes carbon dioxide? This question has been following since from the last few lectures. So okay, so chemosynthetic lithoautotrophs. So because they are autotrophs, they fix carbon dioxide, and litho in the sense they oxidize inorganic compounds to derive energy. So chemosynthetic lithoautotrophs can oxidize inorganic compounds for energy, and they can fix carbon dioxide. So we can just break down the name itself and then answer all questions that can be of this type. So in natural habitats, bacterial population growth is typically uh, which of the following? Whether the growth is exponential or sporadic, dormant or unaffected by resource fluctuation. So in natural habitats, bacterial population growth is sporadic or slow. So which of the following is not a direct selective pressure confronting microorganisms? So nutrient uptake regulation directly provides a selective pressure. Competition and predation is exactly what shapes the community dynamics, and chemical fluctuations can also be a selective pressure, whereas genetic mutation rate is not a direct selective pressure for microorganisms. So out of the following statement, identify the true statement. Both B and C are right answers. Substrate level phosphorylation is not exclusive only to glycolysis. It happens in other metabolic pathway like TCA cycle also. And all carbon fixation is in nature is not light dependent. There are also light independent carbon fixation pathways available. So proton motive force is essential for ATP synthesis via ATP synthase. Yes, um, proton gradient is the essential driving force for ATP synthesis. So that uh, potential proton difference is called the proton motive force that drives ATP synthesis. And free energy gain from reducing oxygen to water is higher than for other acceptors. So last week we discussed about this redox tower. So when we are seeing the redox tower, the energy gain from reducing oxygen to water is very much higher than other acceptors. In continuation to that question, we have this question. Oxygen reduction is most energetically favorable reaction in biological systems because free energy gain from reducing oxygen to water is higher than for other acceptors. So we know that the reason is true. So I think yes, this is a repetitive question only. So both A and R are correct because the energy gain is more. It is most energetically favorable. So the next question is microbial gene regulation allows simultaneous use of multiple electron acceptors. This maximizes cellular ATP production. answer is B. Uh, Microbial gene regulation does not allow simultaneous use of multiple electron acceptors. So whichever is the most favorable, that is used first. So when oxygen is present, oxygen is used first, followed by some nitrate or sulfate based on the presence. It does not allow simultaneous usage. Uh, and this phenomenon itself is not present, therefore this is not maximizing cellular ATP production. Again, a question on identifying the true statements. A statement is methanogenesis readily occurs in highly oxidizing environments. All environmental systems rapidly reach thermodynamic equilibrium. Fermentation is energetically preferred over aerobic respiration if oxygen is present, and photosynthesis produces both oxygen and reduced carbon molecules. So methanogenesis readily occurs in highly reducing environments and not in oxidizing environments. All environmental systems do not rapidly reach thermodynamic equilibrium. They stay in a state of inequilibrium only. Fermentation is not actually when oxygen is present, aerobic respiration route is preferred. Whenever oxygen is not available in abundant or sufficient quantity, then only fermentation is preferred. And finally, the last statement, photosynthesis can produce reduced carbon molecules like glucose and also oxygen. So only the last statement is right. Gene expression for using alternative electron acceptor is inhibited when dash is available. So based on the previous 3 to 4 questions that we have seen, this question's answer. So based on the last 3 to 4 questions that we have seen, this question can be answered easily. Okay, so based on the previous question, oxygen is the answer. So gene expression for using alternative electron acceptors is inhibited when oxygen is available. Only when oxygen is absent, we go for alternative electron acceptors. So I have few slides on the redox tower. So before that, I will just skim through the remaining slides that I have, just 2 to 3 questions. I will not wait for your answers, but just post the answers. So you can see these questions from the PPT that I will be uploading in the drive. So gene regulation in microbes often ensures that only the highest energy yielding pathway is active. So now we come to the redox tower. So as I mentioned in the last class, so this is the redox tower. The image that is present on the right side is the redox tower. So here each redox pair has a standard reduction potential value, which is indicated by this E naught dash. So this E naught dash, or the standard reduction potential value, indicates the tendency of that electron pair to gain electrons. Okay, so the tower arrangement is such that the half reactions from the most negative to the most positive are present from top to bottom. So the top part has the most negative e dash value containing redox pairs, whereas on the bottom you have redox pairs with the most positive e naught dash value containing redox pairs. Okay, and the electron flow direction is such that it will flow spontaneously from electron donor. So the donors are present on the top of the tower, while the acceptors are present at the bottom of the tower, and from the donor to acceptor, from top to bottom, it will flow spontaneously. And the greater the vertical distance, that is from this SO42 minus to this oxygen, will be the highest energy yielding process. Like the greater the vertical distance between the donor and acceptor on the tower, the more energy that reaction pathway will release. Okay, so when it is going to be from this H2S SO32 minus electron pair to this fumarate succinate pair or cytochrome pair, the energy yield will be very very less. So when you have more distance covered in this tower, more the energy production will be there. And this is generally used for predicting energy from metabolic pathway and understanding the electron transport change. So as you can see, oxygen is present in the bottommost part of this tower. So we have seen 4 to 5 questions which talk about the importance of oxygen or aerobic environments. Whenever oxygen is present, aerobic respiration is favored, because we get maximum energy gain from this oxygen electron acceptor, because the oxygen water reduction reduction potential value is plus 0.9 volts. Okay, so this is the concept of redox tower, and when you know have the knowledge about this redox tower, you can answer most of the questions that are in relation to this oxygen as electron acceptor, or ion as electron acceptor, something like that. So next one is about the substrate level phosphorylation. So here you only talk about substrate level phosphorylation. So phosphorylation is the addition of phosphate groups. This is again very important in terms of metabolism, because ATP, NADP, everything is present, and ATP production is the predominant energy generation pathway, and ATP utilization is the currency for energy utilization and metabolism in organisms, right? So substrate level phosphorylation, the source of phosphate is organic molecule, and the energy source is a high energy phosphate bond of the donor, and in prokaryotic it happens in the cytosol. There are two other types of phosphorylation that can happen. One is the oxidative phosphorylation, and another was the photophosphorylation. Substrate phosphorylation is a direct phosphorylative method, whereas in case of oxidative phosphorylation, you have inorganic phosphate molecules, and the energy source is a proton motive force. You can see this in the electron transport chain that occurs in the cytoplasmic membrane of prokaryotes. In case of photophosphorylation, you need light. So whenever some organism has this uh, chlorophyll pigment or something, you can find photophosphorylation happening in that. Like the source of phosphate is inorganic phosphate, and the energy source is the proton motive force. Mostly you can see this in thylakoid of chromatophores. So the next topic is about this glycolysis. So this is the glycolytic pathway. Here substrate level phosphorylation happens. So substrate level phosphorylation, as I told you, directly transfers a phosphate group from high energy substrate to ADP, creating ATP. ADP is adenosine diphosphate. So in substrate level phosphorylation, when there is diphosphate, directly from one substrate, phosphate group is taken and it is added to the diphosphate molecule to create a adenosine triphosphate molecule. So this is the energy currency. So when breaking down this bond, phosphate bond, you get some energy. So that is why ATP is called the energy currency. And in glycolysis process, this occurs twice for each molecule of glucose. So as you all know, until this step that I am pointing out here, it is the uh, breakdown phase, and after this it is the payoff phase. So in the breakdown phase, ATP is utilized, so energy is utilized and ADP is generated. Only after this step, after half of the glycolysis, you can see ATP is generated from ADP, and it also happens in two steps. So one is the step where this phosphoglycerate kinase transfers, this is the phosphoglycerate kinase enzyme, from 1,3 bisphosphoglycerate. There are two phosphates present, it is reduced to 3 phosphoglycerate. So the phosphate group present in the first position is taken and given to a ADP to form ATP. And in the last step, where phosphoenol pyruvate is converted to just pyruvate, so this phospho group is taken from this phosphoenol pyruvate and leaves us with pyruvate, and the phosphate is given to this ADP to form ATP. So directly from substrate, when you are giving it to ADP, it is called substrate level phosphorylation. So substrate level phosphorylation is responsible for generating 4 ATP molecules during the payoff phase, because 2 ADP molecules are given. So as we all know, up to this step we have uh, 1 molecule. So this C6 glucose is broken down into 2 molecules, and therefore whatever reaction happens is going to happen twice. This DHAP is also converted to this GLAP, and everything is going to be twice. That is why we are getting 4 ATP molecules during the payoff phase. And finally, after that, we have this TCA cycle or citric acid cycle, which you can go through. I have not added much questions about this TCA cycle in this, but just you have to know what TCA cycle is. And finally, this is the uh, electron transport chain in prokaryotes. So you can see there is a cytoplasm and there are some transfer systems present here. So what happens is this electron gets transferred from one to another, like you follow the direction of this arrow, and in that way the electron gets transferred, and finally you have the enzyme called ATP synthase. So you can see that from all this redox reaction that happens in the first part, protons are present outside the cytoplasmic membrane, but the proton concentration inside the cytoplasm is very less. So therefore there is a concentration gradient in the sense the proton concentration outside the membrane is high and inside the membrane it is very low. So the only way for uh, mitigating this gradient or the concentration difference is to send protons from outside to inside. So how the protons are sent from outside to inside is through this ATP synthase. So when these molecules are flowing through this ATP synthase, they can convert this ADP to ATP. So that is how this electron transport chain works. So I am just giving a pictorial representation for you to understand all the key mechanisms that are involved. I hope the question answers and the explanations were clear. If you have any doubt, please post in the chat box. Otherwise, you are free to leave the session. Thank you for attending today's session, and thank you for your active participation in all the question and answers. I hope the session was useful. If you have any doubts, please post in the chat box. Otherwise, we can close the session for today. Can can we close the session? Ashwini and Shailaja, can we close today's sessions, or do you have any more doubts? Okay. Thank you for the session. Looking forward to see you both in the next session also. Thank you for joining today. I'm just closing the meeting now. Thank you so much.